now we have so much data around that CAR T can be used upfront in the relapse and refractory setting. That means that when the CAR T was approved, they were approved after the failure of multiple lines of chemotherapy or immunotherapy. And now the new trial data came last year where CAR T are coming as upfront in the treatment. And because of these studies, now many more people can get the access of CAR T. The CAR T not only treat patients for the short-term duration or the induced remission just for short-term, but in fact it can do cure for long-term. And we have seen a lot of cases of long-term cancer cure. And Emily Whitehead was the first patient, actually one of the first patients who was treated with CAR T at UPenn, and now she is basically declared cured. I think that's where academia industry partnership is extremely important. That I think first time happening in the country where academic research, two academic institutions brought up the basic science and now take this basic science to society. That's where ImmunoACT and Lotus Lab, they are helping us to take this academic research to the masses and to the public.
This is a second generation humanized anti-CD19 CAR T-cell therapy with 4-1BB as the co-stimulatory domain. And this is a patented product. The very important finding or observation from the preclinical work was the amount of activation of the interferon alpha, which was happening in the preclinical studies, was actually minimal. And therefore, what was expected was that this will cause lower amount of cytokine production. And then therefore, the hypothesis was that this will be safer to handle in the clinics. To the first infusion, actually, we did not notice a neurotoxicity across a wide range of doses that we used. Also, interestingly, no patients required any ICU admission, because that was the biggest worry when we started the trial, that many of these patients would develop complications and would require ICU care. And most of these toxicities were manageable.
But I think what Rahul's CAR is pretty safe if they are so adventurous, giving so many cells. So we wouldn't want to give higher numbers. But I think if you have given it in a very high tumor burden load and you haven't seen CRS and you still are showing efficacy, I think that's fantastic.
Basically, we all know that the power of lymphocytes, and this talk image from NIH shows you two lymphocytes attacking a lung cancer cell. Lymphocytes have an inherent capacity to destroy cancer. Then why doesn't it do it? And we all know that's because we know that lymphocytes are accumulating inside the cancer. So the cancers do attract lymphocytes. They are immunogenic, but they don't die because these cancer cells develop mechanisms to sort of evade their action. So whether you're engineering the T cell or the NK cell or using phagocytes, whatever you're using, ultimately they all land up at the site and they're all acting in different ways. So when we engineer a T cell and talk about CAR T cells, we are just talking about an initiating mechanism which ultimately leads to the tumor destruction.
So hematological malignancies, now CAR T cells are coming one after another, and myeloma it's there, in lymphoma it is there, in leukemia it is there, AML it may come shortly. So there is a lot of bombardment happening, and it is going to challenge transplant, indications of transplant, maybe no transplants.
I would like to talk about what academia provides. Academia provides talent to the industry. That's the mother of knowledge generation, basic research. And they also generate a lot of ideas which could be through their incubation centers. Like IIT Mumbai, a lot of incubation centers. But industry provides different things. Industry acquire talent pool from the academia. So we don't have any industry to generate talent. We acquire talent pool from industry. And we're good at converting your basic research into advanced science.
Look, then for every CAR in the country, we can do some 4 or 5 centers can monitor that, and then we can decide which patients will go for transplant. And then again, pooling data together from different centers, different CARs.
I think we are set with a regulatory pathway at present. Like we have a couple of products already from the cell gene therapies or other cell-derived products. So we follow the similar pathway right now, which I think we also have a platform with and discussion platform with all the other regulatory agencies, I mean, around the world.
So when we talk about this gene and cell therapy, the few suggestions I have to make is that we should try and engage the key opinion leaders and even the policy makers with regard to fundraising on cancer, and also all the representatives from all these 4 segments to be involved from now and not later on when the drug is approved.
We have empaneled hospitals across the country where such kind of treatments can be given, because the patient is traveling, again the out-of-pocket expenses will go very high. So these are some of the ways which ICS has been using, the philanthropic funding, the CSR.
And then once your scale improves, the cost will come down to a certain extent. And also a lot of international patients will come. That will be a beneficial to the country as well as the hospitals and the whole ecosystem.
Talking about scalability, more about horizontal scalability, we already spoke about, we don't want to be a one city, one place, centralized manufacturing setup. We want to go close to where the patients are. And as India, we have proven success in multiple high-scale complex goals, right? Be it the Mars mission or the UPI, the startup. So there is no reason why we cannot achieve a global platform, a made in India global platform for cell and gene therapy. That remains our ambition. So I hope that sort of gives a perspective of where ImmunoACT is and where we want to be basically.